Assalamualaikum warahmatullahi wabarakatuh. Hello everybody. I would like to say thank you for watching my presentation. Introduction, my name is Wahyu Susanti with the list number ID 027. I'm from Biology Education B, Semarang State University. Today I'm here going to presentation about urease test on enzyme. The urease enzyme is an enzyme that plays a role in catalyzing the hydrolysis reaction of urea into ammonia and carbon dioxide. Urease is commonly found in a variety of organisms, including plants, animals, and microorganisms. One of the natural sources that are rich in the enzyme urease is soybeans. In soybeans, urease plays an important role in converting urea to ammonia. In carrying out its activity, enzyme can be influenced by several factors such as substrate, temperature, pH, cofactors, and inhibitors. The purpose of the urease test is to prove the presence of the enzyme urease in soybean suspension. This experiment uses tools in the form of 3 test tubes, a water bath, and a pipette to measure the volume of the solution. In addition to tools, the materials used in the urease test include soybean suspension, 1% urea solution, phenolphthalein solution with a pH of 8,3 until 10, and 1% HgCl2 solution. The way this urease test experiment works is, first prepare 3 clean and dry test tubes. So, place the following solution in a test tube using a pipette. Test tube 1. Pour 1 ml of 1% urea solution, add 10 drops of soybean suspension, and add 2 drops of phenolphthalein solution. Test tube 2. Pour 1 ml of 1% urea solution, add 10 drops of hidden soybean suspension, and add 2 drops of phenolphthalein solution. And test tube 3. Pour 1 ml of 1% urea solution, add 10 drops of soybean suspension, add 2 drops of phenolphthalein solution, and add 10 drops of 1% HgCl2 solution. After that, mix the solution in the tube by stirring. Especially, place the tube in a water bath at 37 until 40 degrees Celsius for 5 minutes. The last step is observe the change in color of the solution after 5 minutes. From this urease test experiment, several results were products including test tube 1 show a pink color. So test tube 1 is positive for the enzymes urease. Test tube 2 show white color, so test tube 2 is negative containing the enzyme urease. Test tube 3 show a light orange color, so test tube 3 is negative containing the enzyme urease. This experiment used the identification method, namely the urease test. This urease test is based on the principle of enzyme action by yield and change that occur in the urease substrate and phenolphthalein solution in the test. Tube 1 soybean suspension was positive for the presence of the enzyme urease, resulting in a change in the color of the solution to pink. This shows that the enzyme urease is active in breaking down urea into ammonia. Then this urease enzyme will increase the pH of the solution and change the phenolphthalein to pink. Tube 2, the heated soybean suspension was negative for the presence of the enzyme urease because it saw a white color. This occurs because heating at high temperatures causes denaturation or damage to the urease enzyme. As a result, the enzymes are no longer active and cannot decompose urea into ammonia, so that the color change of phenolphthalein does not occur. Top 3. Soybean suspension with 1% HgCl2 was negative for the presence of the enzyme urease because it saw a light orange color. This happens because the enzyme is inhibited by 1% HgCl2 solution so that urea cannot be broken down. This inhibitor prevents the enzyme urease from breaking down urea so that the color of the solution becomes light orange, not pink. Based on the result and discussion that has been described, the conclusion from this experiment are in the urease test it can be interpreted that the soybean suspension produces ammonia which is alkaline, so that the pH of the solution increases, which when added phenolphthalein in will turn pink in test tube 1. In the heated soybean suspension, the urease enzyme will be damaged and the addition of 1% HgCl2 solution because it functions as an inhibitor that inhibits the performance of the enzymes to the result negative. I would like to thank the laboratory assistant who helped me in carrying out the practicum and provided a lot of information during biochemistry lesson. That was my presentation. Thank you for listening my presentation. I would like to apologize if there is a mistake. I hope you have a nice day. Wassalamualaikum warahmatullahi wabarakatuh.